Crystalline water keeps coming and going in a stream of...
I've spent my entire adult life thinking about medicine and biomedical questions from the conventional reductionist approach. And in graduate school, we focused on very well-controlled experiments, as most graduate students do, only varying one parameter that we wish to point our attention toward in understanding a mechanism. And similarly, trained as a physician, I focused my attention on the minimalist approach to defining a disease. And just as William of Ockham, the famous logician from the 14th century, pointed out that plurality should not be focused on except when absolutely necessary in describing a disease or a disease mechanism. And we as clinicians take that dictum to heart. So that most of the complex diseases we deal with in adults, diseases like hypertension or atherosclerosis or cancers of various sorts, while these are extraordinarily complex biological phenomena that underlie them, we strip them down to a bare minimal number of qualities or clinical features that allow us to name each of them under a very broad rubric. So for example, anybody who has an elevation in the pressure measured in an arm with the cuff above a population mean is considered as having hypertension. But hypertension is really a syndrome that comprises many different diseases of many different causes. And this kind of conventional reductionist approach, while it makes our lives easier as clinicians and diagnosticians and therapists, it really eliminates much of the nuance of the biology underlying complex disease. And more important, it limits our ability to tailor therapies in a personalized way to individuals with these complex diseases. An example from an area that I know something about the basic biology of to highlight this point. There is a metabolic disease that can elevate an amino acid in the blood called homocysteine. Homocysteine is a sulfur-containing amino acid that's related to cysteine in the sulfur metabolism pathway in mammals. It was shown by McCulley now in the 1960s that individuals who died of a rare inborn error of metabolism called homocystinuria, in which the levels of this amino acid in the blood are about 10 to 20 times normal, that those who died with this disease in their 20s or 30s had extensive vascular disease, premature vascular disease. It looked like atherosclerosis, but not quite. And McCulley, exploring and examining those tissues and seeing what he noticed as narrowed vessels, proposed that mild levels of hyperhomocystinemia, not these very severely elevated levels, which are more prevalent in the general population, could also contribute to garden variety atherosclerotic disease, heart attacks, strokes, peripheral arterial disease. As a result of that hypothesis, over the next 30 years, there were many epidemiological studies that supported that notion. When population distributions of homocysteine were compared against the prevalence of vascular disease in that population, there was a pretty good correlation. And as a result, the community of clinical investigators who are focused on reducing the risk of atherosclerotic disease considered ways by which one could reduce homocysteine levels in the blood as a therapy that could be preventive. Now, homocysteine's metabolism is a little complicated, but one simple way to reduce levels of homocysteine is to facilitate its remethylation to methionine. That remethylation process is governed by two enzymes, one of which requires folate, folic acid, and vitamin B12. Given that well-known mechanism, there were several large clinical trials that were designed to address the following question. Could simple treatment with folate reduce homocysteine levels in blood and at the same time reduce the risk of clinical events in patients with established vascular disease? So these are so-called secondary prevention trials. And these studies began in the late 1990s, continued to the mid-2000s. And all of them were negative. They all failed to show any benefit on clinical event rates, despite the fact that homocysteine levels were significantly reduced by the folic acid treatment. So the community of clinical investigators in vascular disease gnashed their teeth and pulled their hair and asked the question, how could we have been so wrong? This is a simple hypothesis. Folic acid facilitates the remethylation of homocysteine. Homocysteine levels were lowered by the treatment. It's a simple, safe treatment. Why was there no benefit to clinical events? And as I thought about this problem and wrote a commentary on these trials, I realized that from a rather simple perspective, what we failed to take into account is that folic acid has many other effects beside lowering homocysteine. It is, of course, an important determinant of DNA synthesis, of cell proliferation. In fact, the earliest cancer chemotherapy agent was a folic acid antagonist that Sidney Farber used across the river at Children's Hospital in the 1950s to treat a form of childhood leukemia. So we know that folic acid influences many other reactions. Another large class of reactions that it can influence are reactions that involve methylation. Folic acid is necessary for the genesis of the major methyl donor in mammalian cells, which is S-adenosylmethionine, derived from methionine, which in turn is derived from homocysteine by the action of folic acid. And methylation reactions are rather broad and large in number in mammalian cells, one large subgroup of which are methylation reactions that govern DNA epigenesis, govern DNA transcription. And the methylation of CpG-rich islands in the promoter regions of genes, as you are aware, will shut off certain genes, important in development and also important in stress responses and many different disease mechanisms. So given that multiplicity of effects of folic acid, the question shouldn't have been, why didn't folic acid work in these trials? The question should have been, why did we think of using it at all to begin with? And in fact, there was one, during the course of these several clinical trials, there was one small coronary angiography trial conducted in Switzerland in which patients with established coronary disease documented at coronary angiography were given folic acid or placebo, and they underwent a second coronary angiogram, this was after an angioplasty, to look at the rate of regrowth of the plaque. And what was found in this study was that those who got folate had a more robust regrowth response than those who got placebo, consistent with folic acid's effects in cell proliferation in the smooth muscle cells of the injured artery wall. This example of a clinical trial that focuses exclusively on a single drug target with a single mechanism of action in mind really derives directly from one of the founders of the field of pharmacology, namely Paul Ehrlich. And as you remember, Ehrlich developed or identified a single agent that killed the spirochete and treated syphilis. It was also a useful agent at treating trypanosomiasis. And he did it by what we would now call phenotype screening. He basically took small molecules off his chemical shelf and tested several hundred of them. And when he got to compound 616, which turned out to be arsphenamine, salvarsan, turned out to be the first treatment for syphilis. And he defined the term in his discussion of that finding of magic bullet. And pharmacologists since the 1930s and every major pharmaceutical company since that time has focused its attention on identifying magic bullets for every disease, no matter how complex, and no matter how complex the molecular network within which the target sits that governs that disease. So a right-thinking person, even if one wasn't aware of the elegant science around networks, network structures, network dynamics that have evolved from many groups, and I know you've been part of one of these as well yourself, even without a knowledge of the details of that elegant science, a right-thinking person on a moment's reflection would have to realize that if you are looking at a collection of molecules that are linked one to another by a series of enzymatic reactions or simply binding interactions, looking at that population of different molecules, one would be hard-pressed to argue that affecting only one of them could uniquely affect the phenotype that you're interested in changing. Given that history, I began to think deeply about how one might redefine human disease. And I have a lot of experience about thinking of how one defines human disease. I'm one of the senior editors of a major textbook in medicine that most students learn from, Harrison's Principles of Internal Medicine. So I know a lot about how disease is classified. And to give you a little background, the classification strategy we use for human diseases really derives from the early work of Malpighi and Osler in the 19th century. And Malpighi was a microscopist who tried to correlate abnormalities in histologic patterns with disease, well before disease was really clearly classified as Osler classified it subsequently. And Osler was the father of the concept of clinico-pathologic correlation, which means that a person who presents with a clinical syndrome, fever, cough, the production of green sputum and shortness of breath, that syndrome correlates with a tissue finding at autopsy, namely an infiltration of the lung tissue with lots of inflammatory leukocytes, and subsequently the identification of microorganisms that contributed to that inflammatory process, i.e. pneumonia. So that link between pathologic abnormalities and clinical syndromes was truly unique and groundbreaking at the time at which Osler's methodology evolved. It was very reductionist. It was consolidative. Many of these syndromes appeared to a clinician to be common one to another. And it was only as our pathological skills got more and more refined that we were able to dissect the subsets of these different diseases. A fine example of this in the modern era is how one categorizes the different kinds of lymphomas. They can all present with lumps and bumps and collections of lymphocytes that don't belong in the places in which they cause the lumps and bumps, but they're clonal, they proliferate clonally, they have very different markers, and one can categorize various subclasses based on this molecular identification of markers that one could never have conceived of by the simple clinical presentation of these patients. So the Oslerian approach, reductionist as it was, really began to facilitate the development of the true scientific basis of medicine in the late 19th century. But it honestly hasn't changed a whit. It's still reductionist, and while we're getting better and better at identifying markers that segment diseases into categories that become increasingly individualized, we haven't gotten that much better at treating many of these common diseases. And in parallel with that development, we have the incredible development of the pharmaceutical industry up through this era, which is to say, beginning as an industry that focused on phenotype without knowing about molecular target, and simply giving drugs that could influence a physiologic effect, like blood pressure, or pulse, or respiratory rate. And identifying those drugs that could have the desired effect, those agents that could have the desired effect with a minimal toxicity. That kind of phenotype screening was the strategy with which the industry began. And what matured the industry at that phase, of course, was the development of organic chemistry and bioorganic chemistry and its application to pharmaceuticals. Through the middle part of the last century, that was the strategy. Toward the end of this last century and up to the current time, of course, the difference has been now we can identify the targets that these drugs attack. So target identification is a major focus of every major pharmaceutical company. No matter how complicated the disease, what you have cancer type X, there appears to be an activation of a certain kinase in that tissue. Tell me which kinase it is. Let's look at its tertiary structure. Does it have an element of tertiary structure in the total protein structure that provides a binding site for a small molecule that could influence the kinase's activity? If it does, then let's examine and explore the effect of that small molecule on a cellular model of the disease, and then an animal model of the disease, and then a patient with the disease. Now, you could say that if this strategy were as effective as the industry believes, that we should have an explosion, an absolute explosion of effective drugs approved by the FDA that should be increasing exponentially as the encyclopedic pool of definable proteins whose crystal structures are increasingly known become more widely identified. But in fact, it's just the opposite. If you look at the number of drugs the FDA has approved, they've decayed miserably in the last 10 years. Up to the mid-1990s or so, the FDA was approving between 50 and 100 drugs a year. In the last 10 or 11 years, they've approved between 23 and 27 drugs a year. A little bit of a blip this past year, but still much lower than had been approved before that. So, you can ask the question then, if we're this much smarter about identifying targets, understanding their structures, using in silico screening to at least theoretically define small molecules that could affect the action of those proteins, testing them in models, why have we not been better at treating complex disease and getting approval to use these drugs to treat complex diseases? Well, if you ask a pharmaceutical executive this question, they'll likely make some of the following comments. Well, we've run out of all the obvious targets. The diseases we're looking at now are increasingly complicated. They will also say that the regulatory environment has become almost prohibitive in many respects, that the strategies that are necessary to achieve FDA approval are becoming increasingly complicated and increasingly expensive. And all of that's true. I don't doubt that. But I think that one of the major reasons that the pipeline is as dry as it is and the FDA approval rate has remained low despite this huge increase in the knowledge base of drug targets is that we continue to think about these targets in reductionist isolation rather than in the context of the network in which they work.
Crystalline water keeps coming and going in a stream